Hello everyone, my name is Ujwala and welcome to the channel Sense. In this world, we all are animals. We are social animals, other are forestic animals and many more. So all of us who contain body, our body is made up of a unique structure called cell. This cell is present in each and every living organism. But the major constituent for the cell present inside the nucleus, that is majorly DNA and RNA. If we talk about the DNA, that is deoxyribonucleic acid, it is a double-stranded helical structure which is more complex than any other structure of the body. And if we talk about RNA, that is ribonucleic acid, is a simple single-stranded structure. So today we are going to focus on this RNA. Basically there are three main types of this RNA. The one is a mRNA, which is messenger RNA. It is a single-stranded and long structure. Another is rRNA, that is ribosomal RNA. Because of the ribosomal structure with it, it is called ribosomal RNA. And third one is tRNA, that is transfer RNA. Contain various loops inside the structure, so it is little more complex. But the major function for all this type of RNA is this protein synthesis. Now let us focus on the first one, that is rRNA, that is ribosomal RNA. Here it is constituent of ribosomes. So the name contains ribosomal RNA. Here it is a site for protein synthesis because at the time of protein synthesis, that is translation, the mRNA will join with the rRNA, but the major joining portion is with ribosome. The ribosome contains two subunits. One is a large subunit, another is a small subunit. Here, if we see in detail, this ribosome is different in both the cases, whether it is in a prokaryotic cell or organism or eukaryotic organism. In case of prokaryotic ribosome, it contains two subunits of 50S and 30S, while in eukaryotic ribosome, it contains basically this 60S and 40S subunit. And combinedly, both of these ribosome makes complete 70S unit for prokaryotic and 80S unit for eukaryotic cell. So this is how this ribosome is different in both prokaryotic and eukaryotic cells. Go further in the tRNA structure, that is transfer RNA. It looks like little complex loop forming structure, which is also known as double RNA or sRNA. It is total 50% of the RNA numbers in the whole cell of the body, and it is majorly hundreds of types are there in the cell. This tRNA basically present in the cytoplasm of the cell, that is the surrounding area of the nucleus. It is also considered as the smallest RNA because the long strand of RNA converted into loopal structure, and because of these loops, its compound makes a group-like structure. and so it is considered as a small structure. It carries amino acids to the ribosomes during translation of the protein, and then it allows over there on the mRNA to form a new amino acid structures. Here it is a single, but as we can see over here, it is a folded structure on itself, which makes it a clover leaf-like form. It is in a 2D dimension. If we observe in a 3-dimensional, that is 3D, it looks like a small L-like structure. Now, if we see in detail about the structure of tRNA, it contains a topmost area, that is amino acid attachment site at the 3' end of the strand, where the amino acid will be bind at the free OH molecule, and then after it carry to the mRNA for the translation purpose. Other arm is a ribosomal binding loop. It is also known as T-Psi-C arm. Other than that, at the bottom there is an anticodon region, because here 3 codon numbers are there, which is called anticodon, because when it attach with mRNA, they contain another sequence known as codon. So both are parallelly attached in a complementary form, and then the protein synthesis can be begun. Another arm at the other side known as amino acyl synthetase binding loop, because at the time of protein synthesis, that is translation, this amino acyl synthetase will bind over here. It is a enzyme for the translation purpose. This is also known as D arm. After that, as you can see over here, the strand is binding internally with itself with the hydrogen bonds, which is between all the pair bases. And one more small structure, that is a small arm known as variable arm. Sometimes it may not be present because it is an extra arm. Afterwards, if we move to the another type, that is mRNA, known as messenger RNA. As per its name, it contains the message from the DNA, because it is directly synthesized from the DNA from nucleus. Then after it come out from the nucleus in the cytoplasm for the translation purpose. It is considered as the longest RNA type, and it is 5% of total RNA of the whole cell contained. It is also contain a genetic information from DNA, as you can see over here. Because of this genetic information, it is considered as containing genetic code, which is called codon. So, at the time of translation, when tRNA will come and bind to this mRNA, that will contain anticodon and the mRNA will contain codon. So both will be complementary bind to each other and translation can carry forward. Then after, there is a starting area on this mRNA called methylated region, because it is coded as AUG, which is typically code for methionine, that is the amino acid. This is an attachment with ribosome. At this side, the ribosome will be attached because it is called as an initiator or a start codon. From here only the protein translation can begin. Afterwards, this AUG, the tRNA will come and bind along with ribosome, and then the translation will begin after AUG sequence. When it began, there is other sequence which is in a parallel and following form. It is known as coding region, which is a simple protein synthesis unit. But then after the end portion called terminator codon, they may be UAA or UAG or UGA. Any of this can be present at the end of the mRNA, which leads to terminate the translation, that is the protein synthesis. Because of this region only, the termination can be possible. So this is how the whole mRNA structure contains three things, that is initiation, then coding region, and the last terminator codon. And the whole mRNA will help for the protein synthesis process. This is how the RNA is considered among these three types, that is messenger RNA, a single-stranded long structure, ribosomal RNA, that is contained along with ribosomal subunits, and the transfer RNA, which contain amino acid on it, so it can bind to the messenger RNA along with ribosomal RNA, and the protein synthesis can form easily and with better conformation.